Micro RNA is a small non-coding RNA molecules, and it's made up of 20 nucleotides. And these micro RNA are found in plants, animals, and some viruses. And the main function of this micro RNA is involved in gene silencing, that is, or in RNA silencing. That is, when we said RNA silencing, it means that this micro RNA can be used to silence or to stop the translation of messenger RNA into proteins. And that is the main functions of microRNA, is to block the translation of proteins. That is to inhibit the conversion of mRNA into proteins. And also it's involved in post-transcriptional regulation of gene expression. And remember, I told you gene expression is like the production of a particular proteins, either in small quantity, in higher quantity, or in a desired amount at a particular period of time. So the expression of the genes, it means the production or the synthesis of proteins from the messenger RNA at a particular period of time. And this micro RNA resembles small interference RNA. And remember that RNA interference is of two types, which is micro RNA and then small interference RNA. So they resemble each other. And one very interesting thing is this micro RNA is usually inhibits the mRNA, or it inhibits the translation of the proteins at the level of the cytoplasm, that is at the cytoplasm. Remember that usually mRNA is synthesized in the nucleus and transported to the cytoplasm where the protein synthesis is taking place. So the RNA or the microRNA blocked the synthesis of these proteins in the cytoplasm, thereby inhibiting the protein synthesis. And these two proteins, this RNA, which is microRNA, and the small interference RNA play the same role. And one microRNA, or one particular microRNA, can inhibit or can block the silencing of thousands of messenger RNA. So it means that one messenger RNA, one microRNA can inhibit the functions of multiple messenger RNA. And then the structure of the messenger RNA is double-stranded. It's made up of antisense, which is known as guides, or the sense strand. And the sense strand is known as a passenger, and it's made up of 20 to 24 nucleotide base pair in length. And they are usually from a hyphen with a loop. It also have an overhang at 3 ends, at 5' ends, and also at the 3' end. So it means that on the structure of the microRNA is double-stranded and is made up of 20 to 24 nucleotide base pair, and then it's also made up of a hyphen, and it has overhang. And now, the synthesis of the microRNA. The microRNA also have its own gene, and this microRNA is translated, or this microgene is transcribed to produce pre-microRNA by the use of an enzyme called RNA polymerase II in the nucleus. And then another group of enzymes called Drosha DGCR8 complex will now cleaved the hyphen, or will now clip the messenger RNA to produce the precursor microRNA. And this precursor microRNA is then exported from the nucleus to the cytoplasm using a transporter. and this transporter is called exportin 5. And then the dicer will now act on this precursor microRNA to digest it into a smaller, or cleave it into short double-stranded microRNA. And then these double-stranded microRNA will now form a complex with RNA-inducing silencing complex. And this RNA-inducing silencing complex will now recognize a specific messenger RNA, binds to it, and cleave it into two. And the mechanisms of the mRNA silencing is through the cleavage of the mRNA strand into two pieces, or by destabilizing the mRNA by shortening its polyether, or reducing the translation of the mRNA into proteins. So the microRNA is deactivating or inhibiting protein translation, one, by the cleavage of the messenger RNA into two pieces, and then the cell will now recognize it as an abnormal mRNA and then digest it to inhibit protein translation. That is number one. And the number two mechanisms of microRNA silencing is through destabilization of the messenger RNA by shortening its polyether. Polyether in messenger RNA is protected from the exogenous or endogenous exonuclease enzymes. So therefore, once the polyether is being shortened, then the exonucleases can easily chop up and break down the messenger RNA, thereby inhibiting the translation of the protein. And then the third mechanisms of microRNA silencing is through reducing the translation of the mRNA into proteins. So the genes, as I explained, the genes are there in the DNA, which is specifically the introns from the DNA is then transcribed into primary microRNA. And this primary microRNA is made up of 100 to 120 base pair. So the transcription of these genes into primary microRNA is done by an enzyme called RNA polymerase II. And then in the presence of BRCA and DGCRH, it forms now a free microRNA by cleaving the loop in the hyphens, making it to form a double-stranded microRNA. And then now the free microRNA is now exported from the nucleus to the cytoplasm via a transporter protein called exportin 5. And then this free microRNA is silenced by an enzyme, is spliced by an enzyme called dicer. And these enzymes form a matured microRNA. And then the mature microRNA will now hinder the translation of the protein. So what are the applications of this RNA interference? The RNA interference have the following application. One, you can use the RNA interference for testing hypothesis of a gene function. Assuming now you have a particular gene and you don't know the function of that particular gene in a cell. So you can design a specific RNA interference that can inhibit the translation of that particular mRNA into proteins. And then from there, we'll now start monitoring the activity of the cell. And what you observe from that cells will tell you that it may be as a result of the silencing of the gene that is responsible for producing a particular protein. And then second, a target validation. Like for example, if you want to design a drug or vaccine, in drug design, maybe you have an idea that, okay, this particular protein is essential for the survival of a particular parasite. So you can target the messenger RNA of that particular protein, silence it, and see if the microorganisms or if that particular parasite will survive. And then another functions or applications of this RNA interference, you can use it in pathway analysis. Most of the pathways, there are in a particular pathways, you can have a number of enzymes, which are proteins that are responsible for that particular pathway. So assuming now you want to know which of the proteins in that particular pathway that is responsible for regulating or for controlling that particular pathway. So you can use RNA interference to inhibit the genes. Maybe you can start, let's say in the pathway you have 10 proteins. So maybe you can start from protein A, design RNA interference, inhibit it, and then you monitor that particular pathway. Then you move to B, C, and D. And then from there, you will be able to understand how those proteins are important in that particular pathway. And then the number 4, which is the gene redundancy, functional screening, and small interference RNAs can use as a therapeutics. Like for example, in a cancer, there are some proteins that are responsible for the development of cancer. Like in a situation where you have like truncated genes or truncated proteins, most of the truncated proteins, they are the proteins that lead to the production of unwanted cells. If these unwanted cells are not eliminated, then they will be developed and proliferate into a cancer cells. So you can use small interferon RNA to target those kind of genes, inhibit them so that to prevent the production of these unwanted or truncated proteins. So therefore, the small interferon RNAs can be used in therapeutics.